All right, so my name is James Gurtowski. I'm going to talk a little bit about the current advances of sequencing technology. So I'll just give you a little overview of what I want to talk about. I'm going to start with just assembly. So I work for Mike Schatz. I think he talked earlier in the course, and a couple of people from my group have talked. So there will be a little bit of overlap in some of the material I discussed, but I think that you guys have learned a lot, so I think a little overlap won't hurt. But so I'll talk a little bit about assembly in general. I'm going to talk about PAC biotechnology and some of the algorithms we've developed to work with this data, as well as some of the results from the assemblies that we've done. In addition, I'll talk about Oxford Nanopore and the technology, as well as some of the algorithms we've developed and the results that we've gotten from this technology assembly-wise. So let me just first talk about assembly in general. So as some of you probably know already, coverage is very important for genome assembly. We're not reading the genome off in one large read. Instead, we have to sample parts of the genome. And in order to sample the entire genome based on Poisson coverage, we have to oversample it. And we get curves like this when we talk about like the expected contig length for different read lengths, which are the different colors here. As read coverage increases, in general, our expected contig length also increases. This is an imperfect world. In general, there's a lot of other factors involved, like error rate and things that affect this. But in general, we can see that higher coverage is better because we have to oversample the genome to make sure that we've sequenced every base. Another important factor in genome assembly is the read length. Read length helps us span repeats. So when we are looking at assembly graphs, we have something like this where you have a contig that's going into some kind of sequence that's repetitive. So the sequence is found in multiple places in the genome. And so another place in the genome will also be connected to this repeat sequence. So you'll build up a graph like this. And what happens is when you try to reconstruct the genome from a graph like this, it becomes ambiguous, right? You can't tell whether, if you're working on this contig, whether you should go to this purple one or to this red one. It becomes difficult to tease that apart. And long reads really help us with that because you can span these repetitive regions. So in this read, you'll have this repeat region, but you'll have flanking sequence that is unique, so it allows you to unambiguously reconstruct this genome. So prior to having long reads, we used to try to conquer this using like mate pair technology, using Illumina data. So you'd have a long insert, but you only sequence the ends. This kind of technology comes with its own issues that have been solved by longer reads. And now when I talk about long read technologies these days, these are generally single molecule technologies like PacBio and Oxford Nanopore. And one of the trade-offs you have with that is many of these technologies have very poor per base quality. So the error rate is much higher. So if you're used to Illumina data where you have like a 1 or 2% error, the error rate on long read single molecule sequencing technologies is generally much higher. So this is generally not as big a concern because if your errors are random and you have many reads that stack up over the same position, you can generally tease apart what are errors and what are actually true nucleotides. So error rate's generally not such a big issue if you have enough coverage. So just to outline the assembly complexity problem, you have this cartoon view of a genome where you have these unique regions A, B, and C, but there's these repetitive sequences which are identical. So the sequence, it doesn't matter, but just as a cartoon, these repeat sequences are the same. So if you have some kind of short read technology that can't span these repeats, what you get is you try to construct an assembly graph and you get something like this. So you have the reads coming from this unique A region. So you create a contig A, and then you see these reads are overlapping this R. And so you can draw a line to R and you say, okay, well, I have this contig R, which overlaps B. So you create a line here, but then B is going to come back into R. And then R goes into C, and then C goes into R. So you have this very ambiguous graph. And just from this graph, which is how you're trying to reconstruct the genome, you can't tell whether you should go AB, ARB, ARC, or some number of times around this graph. And so long reads really help you with this because you'll have reads that span from A to B with these unique flanking sequences on either end, which make reconstruction much easier. And then, so yeah, you would have an assembly graph like this with these longer reads, which you can reconstruct the original genome exactly. So another important factor that I'll discuss is like how do you measure like how good of an assembly you have. And the common metric for this is N50. N50, it's a single number metric. It's a very imperfect metric, but it gives you like a general idea of how to assess an assembly. And the basic idea is you have, say, an expected genome size of 1 megabase. So this is some hypothetical genome of 1 megabase. And you've done an assembly and you've created contigs. And what you do is you sort the contigs by length, so from largest to smallest. And you just kind of go along the genome and you say, okay, how much of my genome is covered in contigs of at least this size? So in this case, 50% of our genome is covered in contigs of 30 kb or greater. And that's the N50 metric. And it's just a very simple number, but it gives you a lot of, it gives you an idea about a lot of information. So you can kind of say how contiguous is the genome, which is important because the more contiguous your genome is, the better resolution of genes you have and their context. So if you have an assembly, which is just a file it's a huge FASTA file with a bunch of sequences in it, and you have 2 genes in different contigs, you don't really know their relative position in the genome, but all of a sudden, if you can combine those 2 contigs into one contig, then all of a sudden you know that these 2 genes are much closer together and could be regulated in some mutual way of some sort. So this is important for like transposons and other complex sequences, and just getting an idea of how the genome is organized. So that's kind of what I wanted to talk about as far as like assembly, and that's like a good background because that's what I'm going to focus on in the rest of the talk, because this is what Mike Schatz has really focused on in his lab. So like I was saying, long read technology is really important for genome assembly, and so we focus a lot on these single molecule technologies like PacBio. I think you guys have kind of heard this before, but PacBio is this huge instrument. It has these small flow cells, so they're very small in comparison to the whole instrument based on how the technology works. And each one of these flow cells has these small little zero mode waveguides, which are just these holes. And in those holes, you have a polymerase with a DNA strand that goes through this polymerase, and fluorescently tagged nucleotides are incorporated while a camera watches this process. And so what you get is something like this. As the polymerase is incorporating these fluorescently tagged nucleotides over this period of time, this camera watches the intensity of this fluorescence, and the fluorescence corresponds to the base that's being incorporated into the DNA, into the complementary strand. So there's a lot of cool things that you can do with this technology. For instance, adding these barbell adapters allows you to sequence one side of the DNA strand, and then loop around and then read the other side. So what's kind of cool about that is if you get many reads of the same molecule, you can start building a consensus sequence. And this is what's called circular consensus sequencing. And if you have many reads and you can build a consensus, you can reduce the per base error rate. And what you do here is you have a polymerase which has a certain life expectancy. You put a short molecule in there, it'll read around that molecule 5, 6, 7 times, and you build a consensus sequence. And you can get very high per base accuracy, but you're losing kind of the long read aspect of that because you're reading this short molecule many times. So if instead you put a longer insert molecule in there, say like 10 kb, you might only get one long read, which will be very noisy, but it gives you a lot of more like contextual information as far as which bases are closer to which in the genome. So this is the kind of technology we focus on, is these long insert, somewhat noisy reads that you can produce using the PacBio. And so I believe Maria might have talked about this earlier in the week, but we had a project going on in the lab sequencing this SKBR3 breast cancer cell line. And we could just kind of show you the improvements in this technology over the past couple of years. And even in a few short months, our collaborators at the Ontario Institute for Cancer Research were able to increase the yield from 213 megabases per smart cell all the way up to about a gigabase of sequence per smart cell. So only in a couple of months, this technology has improved dramatically. And this is really cool, right? Because the human genome is quite large. So you can see here, this is like a factor of like 5. So you've reduced the amount of sequencing you have to do by a factor of 5, which means that you've reduced the cost of doing this sequencing by a factor of 5, which means that you can take on ever larger sequencing projects in just a few short months. So and this is kind of exciting stuff. So you can see here, like at a gigabase per smart cell, you only have to do, well, you have to do a few hundred smart cells to get enough coverage over like a human genome, but you can start like thinking about like the kind of human-related projects that you can take on, whereas just a few short months earlier, these things were just prohibitively expensive. So looking more at the data that you get off the machine itself. So this is a PacBio read that we have gotten off the machine at some point. And just to kind of look at the error rate, you see that the average error rate with respect to the reference genome is 16% overall. But this breaks down into 30% mismatches, 11% deletions, and 58% insertions. And so our error profile is really dominated by insertions, which is kind of a cool thing because you can kind of hypothesize that as the DNA sequence is going through this polymerase, nucleotides that don't really belong in the sequence or the polymerase is attempting to incorporate them, but throwing them out. The camera can't really tell whether this was an incorporation event or it could have just been like a nucleotide floating around, attempted to be incorporated and was thrown out. So you have these sequences where we have these positions where there are insertions with respect to the reference. And so this is kind of important, understanding the error model, because the algorithms which are going to attempt to correct these reads can use this information to do a better job. And so that's what we've really been focused on in our lab is there have been a lot of assemblers developed going back to about the Sanger days. These assemblers are built on the assumption that the reads are of somewhat low error rate. So in order to incorporate these single molecule reads into those assemblers, you have to kind of do like this pre-preparation of doing this error correction step. And so this is what I focused on a lot in the lab. So there's different ways of doing correction. And it really depends on how much read coverage you have. So what you can do is PB Jelly was developed to try to just fill gaps in scaffolds. So what it does is it takes an Illumina assembly where you have mate pair libraries, and what you get in that is these scaffolds where you have contigs, which are linked together using just in the FASTA file a bunch of ends, signifying basically the estimated length of the sequence that should be there, but you don't know the actual bases. And because these are repetitive elements that the mate pairs are jumping over. So PB Jelly attempts to find these PacBio reads that align to either end of the scaffolds and can combine these contigs. And you usually use this if you have like somewhat low coverage data, because if you have higher coverage data, you can start doing, you can take better approaches such as using hybrid error correction technique. So this is where I focused a lot because this was just kind of the data we had. So I developed this tool called ECTools. There was other tool called PacBio-CA. And what this does is it takes short reads, like Illumina reads, or you could actually give it any kind of reads, or an assembly. And what it does is it aligns the PacBio reads to those assembly or reads, and then tries to do an error correction. So this has been pretty successful for us at intermediary, like between like 5 and 50x coverage. So depending on how much money you have and like what kind of project you're trying to take on, this approach could be very successful. However, if you have a lot of coverage, what you can do is like something called self-correction. So if you have over 50x coverage of PacBio data, what you can do is you can align these long reads to each other. This is a very expensive process, and it takes a long time. However, you do get very good results because you have the ability to align these long reads to each other, which gives you a lot of context for the alignments. And you can then do a consensus of these alignments. And this was developed by PacBio themselves, and it was called the HGAP error correction technique. And this is just kind of a cartoon. You have a PacBio read, you've aligned all these other PacBio reads to that PacBio read, you build a consensus, and then these consensus reads will align to the genome at pretty high identity. And then if you don't have your own genome, you can take these corrected reads and build your own genome using assembly techniques. So I just want to talk a little bit about some of the results we've gotten from using these error correction techniques. So we started out a little while ago with this rice strain IR64. It's a very important crop strain. And we initially had these Illumina data, which we did an all-paths assembly of, and this had, these are pretty expensive libraries to construct with these jumping libraries and everything. We ended up with a contig N50 of about 18 KB, and later on we got some MySeq data. The MySeq reads are a bit longer than the original like 75 base pair Illumina data, and we were able to generate an assembly of about 19 KB. We ran PacBio to CA at the time, and it gave us an N50 of 144 KB. And then, you know, we were trying to optimize this as much as possible, which is then when we developed this ECTools algorithm. And so I'll just talk a little bit about that. The basic idea is instead of aligning the short reads to the PacBio reads individually, we try to take as much contextual information from those short reads as possible. So we do a pre-assembly of these short reads using any short read assembler that you like. And then you align these unitigs to the PacBio reads themselves. And then you can do a error correction based on that information. And the reason we developed this was because we started looking at the alignments of these short reads to the PacBio reads themselves, and we just kind of found that there were these pockets of high error rate in the PacBio reads, and what we were missing was reads that could span across these pockets of high error rate. And so by making these short reads longer, by pre-assembling them, we were able to span these pockets of high error rate and do a better correction. The basic pipeline is we generally use Celera to build some unitigs, and then use NuCMR to align those unitigs to the PacBio reads. Delta filter is this algorithm which allows you to use the longest increasing subsequence algorithm to find the best set of unitigs that span a PacBio read. And then we use show SNPs, which is part of this NuCMR package, to build this consensus sequence and correct the PacBio reads. And the key here is we're not splitting or trimming the reads, because we found that doing that in some of the other error correction techniques, we're doing a lot of splitting and trimming. And this was a heuristic that was developed in the Sanger days that has kind of been messing up the error correction techniques. So just to talk a little bit about the alignment filtering technique. This is based on the longest increasing subset problem. And the basic idea is you have these short read unitigs that have aligned to a PacBio read. You're going to get many spurious alignments just based on sequence similarity throughout the genome and repetitive elements. So instead what you want to do is try to filter these alignments to find the best set of alignments that cover the PacBio read. And then you can just build a consensus of these. So it's kind of like scaffolding the unitigs using these PacBio reads. And so what we ultimately found was that we were able to double the N50 of the PacBio to CA assembly, which at the time was a very good result. So now I'll just go into a little bit more detail about the HCAP error correction. And like I was saying, HCAP error correction does work a lot better than hybrid error correction. That's just basically because you're aligning long reads to long reads. So these alignments have a lot more context. For people familiar with alignment, the longer the read, the more information you have, the more confident you can be in the alignments. Because short reads have a tendency to align many places. So once you've built up these alignments, you can build a consensus sequence, and then the consensus can be very good depending on how much coverage you have, and you can reconstruct the genome using current assembly techniques. So this was just another case study where we were sequencing a yeast. And yeast has a pretty small genome in comparison to some of the other plants and animals that we've sequenced, and we ended up with data that was over 275x coverage using 5 SMRT cells and the P5C3 chemistry, which is a little dated these days, so you can get actually much better data. The mean read length was 5 kb. We had 103x over 10 kb and 7.6x over 20 kb with a maximum read length of 35 kb. So we ran this through the HCAP algorithm, and we were able to produce an assembly that was actually very good. So there was only only 4 contigs that had issues, but well, there was only 4 issues in the assembly, but most of the chromosomes were assembled almost perfectly. We had a overall ID of 99.98% per base error in the per base identity compared to the reference genome, which is really good because there is some difference between the strain and the reference genome, which can account for some of those errors. But it also gives you an idea of being able to take these very noisy reads, build a consensus, and then once you get through all the consensus of the assembler and everything, you can actually take these reads, which started out at 16% error, and get down to extremely low error rate as the final assembly is completed. The only issue we had was this very large repetitive element in the genome, which was over 10 kb, which we just didn't have enough reads to span. At the time, we could have done a lot more sequencing, and I think that even today would be difficult because getting over a 10 kb region can be quite difficult because you have to get lucky with the number of reads that actually span that 10 kb. Going back to IR64, so if you remember, we were doing this hybrid error correction in which ECTools made a big improvement. However, a couple months later, we got a lot more money, and we did a lot more sequencing, and we actually were able to improve the genome tremendously. So overall, we had 115x total coverage, the mean length of 5.9 kb, and over 6x coverage over 20 kb. So this was a lot of data in comparison to what we were doing this hybrid error correction with. And our ultimate assembly results, we had an N50 of 3.9 megabases, so compared to 250 kb before. So the N50 has improved tremendously, and the contiguity has improved by just basically spending more money. Another error correction approach, which is more modern, is using it's called DASCON. It's basically a hybrid between Gene Myers, who has created this D-aligner algorithm, which does very fast read overlapping. So one of the issues with using HGAP in the past was that it used a standard aligner called Blazor to do all of the overlapping. And overlapping is a very expensive process because you're aligning all the reads to all the reads. So when you're trying to do larger genomes, this becomes a very computationally expensive process. It's an n-squared process. So you have, as your genome size increases just a little bit, you end up with a lot more overlaps that you have to compute, which becomes much more computationally expensive. So Gene Myers developed this D-aligner algorithm, which does very fast overlapping, making it possible to do overlaps of vertebrate genomes. And the PacBio guys have created this PBDAG-Kong, which did the consensus algorithm before to work with the output from this D-aligner. And so this is how we've been doing a lot of our assemblies these days. It's a very smooth pipeline. It's just a couple of commands that you can run yourself. If you go to the GitHub page, you can download D-aligner. It has a very good manual. You can learn how to run it. And then at the PBDadCon on PacBio's GitHub, you can download that software, and it'll plug right into the output from D-aligner. So just to give you an idea of, this is an assembly we were working on very recently. It's from this Bumblebee, and so we had a very similar distribution to what we had before. Average read length of about 5 KB. We discarded reads less than 5 KB because we had enough coverage of reads greater than 5 KB that we thought we could do a decent assembly. So using the shorter reads doesn't really help us. It generally just introduces errors. So just to compare, we had this Illumina assembly where we had done this with spades of the same genome, but just Illumina only, which gave us an N50 of 35 KB. And when we ran this DASCON on this PacBio data, we were able to get an N50 of 359 KB. So almost an exactly 10 times greater N50 value, which means the genome is far more contiguous. And actually, this genome did not have as much coverage as some of the previous genomes. So I think this genome is certainly limited by coverage. If the group spent a little bit more money and got more sequencing, especially with the latest chemistry from PacBio, this assembly would improve even more. So that's kind of what I wanted to talk about as far as PacBio. The new kid on the block, as far as from last year, is called Oxford Nanopore, and it's a pretty cool technology. It's a little bit different than PacBio. Instead of having that huge machine that requires like an entire room to operate, this is a USB pluggable device. It's very small. It fits in the palm of your hand, and it's kind of a cool idea because what they're really pushing is this idea that you can use this like in the field, hooked up to your laptop, and you can just sequence random things once you do library prep, of course. But the way it works is it senses changes in ionic current as DNA molecules pass through this pore. So the basic idea is you have this lipid bilayer with a protein pore that's lodged in it. You pass some current through here in an ionic solution, so the ions are flowing through this pore, and then you have like some kind of antagonist that can go through this pore and will create a disruption in that current. And you can see these disruptions in current are somewhat characteristic of like the size and shape of the molecules that pass through the pore. And George Church, like many years ago, I guess like 20 years ago now, has had this idea to use this for DNA sequencing. So this has been in development for quite some time. And the basic idea is you can pass a DNA molecule through this pore and try to sense the fluctuations in the current, which will give you an idea of what the actual sequence of nucleotides is. So it's a cool idea. Some of the advantages of this technology, or what are advertised as some of the advantages, is that it's label-free. You don't have to do much library prep. You just kind of just put the DNA into the instrument. It's single molecule. You don't have to amplify the data. It's pretty high throughput. You have many of these pores in a single instrument, so you're doing a lot of reads in parallel. The instrument itself is very inexpensive, so you don't have to purchase this very expensive PacBio machine, which requires a team to run. You have just this very inexpensive instrument, which you can run personally. The library prep is very simple. So I'm a computational biologist, and I have absolutely no training in the wet lab, but I was actually able to do this prep myself, and it produces very long reads. Some of the challenges that Oxford Nanopore has been dealing with in developing this technology is that it's quite difficult to control the rate of DNA through this pore. So they have developed these motor proteins which sit on top of the pore and kind of feed the DNA through the pore, but it's still very difficult because you're working with these biological compounds that are doing these processes. So it's not exactly perfect, and developing algorithms that can deal with that kind of imperfection can be difficult. Another issue is that the pore does not have a single base resolution. So this pore where the nucleotides actually interact with the pore and create the current fluctuations, it's not the current fluctuations are not at single base resolution. So the signal that you're getting is actually in correspondence to a k-mer. So you generally about 5 bases. So that means that you have 4 to the 5 possibilities for what that signal corresponds to. So the space of possibilities for each current signal is quite large. So decomposing that can be quite difficult. In addition, these nanopores have been used to sequence DNA in the past for actually quite some time in a laboratory environment, but actually commercializing this product is very difficult. And that's mostly because biological pores are very sensitive to environmental conditions. So trying to build this instrument, which you can kind of ship across country or across the world, can be very difficult to keep the conditions proper for like the proteins not to denature and the flow cell to remain at the proper pH and everything. So just a little bit under the hood. So you have this instrument and you have this flow cell, and in this flow cell you have these little channels, and across these channels is where you have that lipid bilayer, and in the middle you'll have this tiny, tiny little pore where the DNA kind of goes through. So here's the DNA prep. It's very simple. People who have done PacBio preps, you'll see that this is far simpler. I think that PacBio prep is a multi-day prep, whereas this just takes a couple hours. And the basic idea is you shear the DNA, you attach a hairpin adapter on one end and a motor protein with like a regular adapter on the other end. And the basic idea is that this motor protein will attach to the pore, feed this DNA strand through the pore. What will happen is you'll hit this hairpin adapter, which will allow you to pull the other sequence through the pore. So you can actually read both what they call template and complementary strand of the DNA. So you get 2 reads of the same molecule, which, similar to PacBio, allows you to do kind of consensus building to improve the error rate. So this is what the software looks like on the desktop. You have kind of a readout of the read length distribution that you've currently sequenced, as well as this kind of heat map view of the pores in action. Which, when you zoom into a different panel, you can kind of see which pores are active, which are not doing anything, and this kind of gives you some immediate feedback of whether your library prep was good or bad, or if you want to. What you can actually do is if your library prep did not work very well, you can actually load more DNA onto the sequencer to try to get a better yield out of that flow cell before the proteins die. So once you get the data off the machine, it comes into your laptop as these H5 files, and these are pretty much just the raw signal data coming off of the machine, and that data gets segmented into what they think are k-mer level measurements. So it segments the raw signal data into these segments, and then uploads that data to the Amazon cloud where all the base calling is done. And this is kind of a cool way of doing things because it allows Oxford Nanopore to continuously be updating their base calling algorithm and such, so that you can always get the latest base calling algorithm by just running the base caller again on your computer. It uploads it to the Amazon cloud, and then you download the sequence data after the base caller is run. So this is kind of what I'm talking about. What happens from the machine, you get this raw signal data, which is sampled at some 10 kilohertz or something like that. And what you're trying to do is segment this into different events, which should correspond to different k-mers that are passing through the pore. And after base calling, you get this sequence of events. The actual base calling algorithm is proprietary to Oxford Nanopore, but they do release some information. It makes sense that it's kind of an HMM, where you kind of settle in on some k-mer sequence based on your event data, And then you can kind of trace through the different possibilities using a hidden Markov model. And the basic idea is that this k-mer will overlap this k-mer by the majority of the bases. It's just you're getting a signal for this 5-mer, but this 5-mer should be only one base off from this 5-mer. So you can kind of build an HMM to guess what the next base will be. So our experience with this last summer was the flow cell yield is highly variable. So in this chart, you kind of just see the dates. And then the red line corresponds to the yield in megabases. And you can see we had like an initial successful flow cell, but then a long period of just these flow cells producing basically no data. And then towards the end, we actually got a few good flow cells that produce upwards of 500 megabases of sequence. And this was in a yeast strain. Overall, you can see, so the purple lines in the background correspond to the read length, and this is the mean, the maximum, and the minimum. And in general, it's about the same because we did the same library prep for all of these sequences. It was just kind of variability on the throughput of these flow cells. So our best flow cell was 446 megabases, but our mean flow cell was only 50 megabases with a mean read length of 6 kb. So this is kind of what the histogram of the read lengths looks like. On the low end, we have a lot of noise. So these are just sequences that are very short, but not very useful for what we want to use this data for, which is genome assembly. We have this spike-in sequence, which is you spike in some lambda DNA, which kind of calibrates the base caller when you upload the data to the Amazon cloud. And that allows you to, it allows the algorithm to kind of just get an idea based on a known sequence what this particular flow cell is doing. So this is where we start to get signal at around 5 kb. This was where we started shearing the data, where our library was sheared to. So you have a mean read length of about 5.4 kb. We had over 41x of reads over 10 kb, 8x over 20 kb, the max read length of 146,000 base pairs. So we had a lot of coverage over this genome, but what we found was that not all of this data was actually very useful to us. The error rate was quite high, so when we aligned this data to the reference genome, we found that actually only 32% of the reads aligned in any capacity. So that means we lost a lot of that data, and it was just very noisy. So we used the BLAST aligner. There's a whole slew of aligners people are using nowadays to align this data to genomes that they claim is more robust, which is probably true in some circumstances. At this time, we found a good parameter set to use for BLAST, which gave us pretty good results. And this kind of just talks about how the difference between 1D and 2D reads. So like I was saying, you attach this hairpin adapter to the molecule. So what happens is you sequence one strand of the DNA, then it goes through this hairpin adapter and pulls through the other strand, and you can build a consensus of those two reads because you know they came from the same molecule. And you can see that this process actually works pretty well. You have here in blue the template reads, which are just 1D reads, with a per base error rate of about 65% identity, so 35% error. But you can increase that by doing this consensus process up to about 75% identity. So that's just kind of the point I'm trying to make here, is that this 2D base calling actually works, and it's kind of a cool way to make the most of the information that they can gain from these reads. So overall, we had about 32% of the data mapping using BLAST-N. We created this chart. It's a little complex, but the basic idea here is on the x-axis we have the read length. On the y-axis we have the percent of the read that has aligned. So that's like how much of this read has aligned to the reference genome. And then the coloring is based on how many reads are in that particular read length bin. So just as an example, here you have full length alignments, right? The full length of the read, 100% of the read has aligned. But these are generally shorter reads on the order of like less than 40 kb. And based on the coloring, 40% of all of the reads less than 40 kb aligned at full length. The other 60% or so, based on color, did not align to the genome at all. As we step further in to the right here, as far as read length, about 40 to 60%, 40 to 60 kb, the reads started to align in segments, so only like parts of the reads were aligning using BLAST, and this kind of makes sense. BLAST is a local alignment algorithm, so you can capture pieces of the read, and it's just kind of showing you that the Oxford Nanopore will have segments of the read that are a very high error rate that you can't really align, but you can align pieces of the read pretty well. And then on the furthest end, you see that these reads that were super long, like 100 kb long, which we got really excited about initially, actually ended up being unalignable to the genome. And we hypothesized that this was kind of just artifacts. Something would get stuck in the pore and just produce reads that were not actually reads of any genomic DNA, but just in fact artifacts. So we wanted to take this data and actually do an assembly. So at the time, the error correction techniques didn't actually work very well with the Oxford Nanopore data, because the Oxford Nanopore data was a much higher error rate than anything we'd seen from PacBio. So we developed this new error correction pipeline, and it's very similar to ECTools, but we called it Nanopore. It uses BLAST as the aligner. And the basic idea is we align short Illumina reads, which have very high identity, to the Oxford Nanopore reads. And then we use the longest increasing subset algorithm to find the best alignments of these short reads to this Oxford Nanopore read and build a consensus. So it's very similar ideas to the previous algorithms, but use different tools in the back end so that we can cope with this very high error rate. And we use PacBio's PBDAGCon, which was developed for building consensus of PacBio reads, but it ultimately worked very well for building a consensus of these Nanopore reads.
So in order to actually build everything, you had to sequence with both the Nanopore and the MiSeq?
Yeah. So at this time we luckily had a library of MiSeq of the same genome available. So since this time, there have been self-correction techniques developed for Nanopore data, but they still suffer from the fact that the Nanopore data has much higher error rate than PacBio, and the yield is pretty low. So they'll work in the publications concerning self-correction of Nanopore data are generally in like microbes and bacterial genomes, which are much smaller than. We did this in yeast, so about 3 or 4 times bigger than some of the bacteria sequences people were using self-correction for. So yes, in this case, we did a Illumina library of the same sample. So you can see that the Nanopore correction does improve the read identity pretty well. So before we had a read length, before we had a mean identity of around 65% ID. After error correction, we were able to bring that up to around 95% identity. So if you're pretty astute, you can see that the histogram of the read lengths has changed quite a bit between before and after error correction. And that's because a lot of data has been lost between the raw, well, I don't know if it's specifically data as in useful data. It's more of a lot of the data, it was not useful because we couldn't align short reads to many of the Nanopore reads based either on error rate or some other problems. So we're losing a lot of data, not necessarily information. We're losing a lot of reads in the error correction step. But you can see we did pretty well. We assembled this yeast, the original assembly from Illumina had an N50 of about 60 kb, and it had, you could just see there's a lot of errors and misassemblies. When we did the Oxford Nanopore, we were able to get N50 of 474 kb. Also had quite a few errors. And then, you know, the kind of champ right now, PacBio, can actually do an assembly of this same strand almost perfectly. So that's kind of just where Oxford Nanopore fits in relative to the other technologies. And this was kind of just an idea of trying to show, like, you know, how you're doing all of these assemblies. Well, so this is kind of justifying our existence in some way. Like, ultimately, you're trying to figure out, like, is doing an assembly, how important is it for me to do, like, an even better assembly than I already have? Like, do I have to spend more money to get a better assembly? And this chart's just kind of showing the functional elements based on the reference annotations of yeast and how many are captured by the different technologies. And we can see when we zoom in on the, on this previous slide, you can see that most of the technologies, even MySeq, can capture most of these functional elements with respect to, like, Nanopore, but only on these longer elements do you see a big difference. And we can see that this long read technology really helps you capture some of the transposable elements, the repeats, the telomeres, which are completely missing from this MySeq data. And so you really kind of want to upfront assess what you're going to be using these assemblies for. If you're really going to be studying, like, these repetitive elements, or you really need very good, like, situational awareness as far as, like, which genes are near each other, like gene context, then doing a long read assembly could be very useful to you. But if you're really just interested in genes and the gene sequence, capturing those using short read technology is pretty easy. So you might not necessarily need to do a super expensive long read sequencing. So just to summarize, single molecule sequencing technologies produce very long reads. They generally have high error rates, which is why we do error correction. But once we do these error corrections, you can use existing assemblers to produce very good assemblies, and they're generally much more contiguous than a lot of the short read sequencing assemblies that we've produced. So on the horizon, some of the future Oxford Nanopore technologies is this Prometheon. So instead of this handheld device, Oxford Nanopore is trying to capture a larger market that try to compete with some of the sequencing, some of the instruments in the sequencing cores by producing this Prometheon, which is just basically a large collection of these small flow cells in one big box, which should increase the throughput dramatically and help Oxford Nanopore compete with some of the existing technologies. And they're even looking to compete with Illumina on that front. The future of PacBio is just recently announced as the Sequel system, which has even higher throughput than the current RS2 instrument, and it's much smaller. It's still not like as portable as an Oxford Nanopore, but the instrument itself is much smaller. So these are kind of just giving you an idea of what's on the horizon. So I just want to thank everyone from my lab, Mike Schatz, Dick McCombie, Sarah Goodwin, postdoc in Dick's lab, who did all the sequencing prep for all of these experiments. I just want to thank everyone in the Schatz lab, Tyler, Han, Hyan, Maria, and Shri. I also want to thank Oxford Nanopore and PacBio for developing these technologies, which we get to play around with. So if there's any questions, let's go. Yeah.